Hi students, so today we are moving on to the next division, that is meiosis. So in the last class we started with a new chapter, cell division and cell cycle. So in this cell cycle and cell division, the division which we studied was mitosis, isn't it? So what was the division we studied? We studied about mitosis. So in mitosis what is happening? In the mitosis you can find one chromosome number 46, a cell containing 46 number of chromosome, that is the human cell. So that cell is dividing and producing how many cells? 2 cells, and all these 2 cells are also having what? 46 number of chromosomes, which means it is an equational division. And what is this division mainly responsible for? It is helping in growth, it is helping in repair, it is helping in increasing the replacement of the worn out cells. Okay, so these are the significance of what? This mitosis. Today we are going to learn about a meiosis, a very important division. This meiosis division, where does it take place? It takes place in the reproductive organs. Okay, so in the reproductive organs, the testes and the ovaries, wherein they are producing gametes, for that this meiosis division is very much important. And this meiosis division takes place in a specialized cells called as germinal cells. Okay, so there only this meiosis division is taking place in the reproductive structure. Now moving on to this meiosis, when you see this meiosis division, what is actually happening in this meiosis division? So the germinal cells, the testes or ovary, whatever it is, there when you are seeing the germinal cells contain how many number of chromosome? 46 chromosomes, right? This 46 chromosome, when it is undergoing meiosis, this meiosis is of 2 stages. First stage is called as Meiosis One and the second stage is called as Meiosis Two. So in this first stage, actually what is happening is that from this one cell which is containing 46 chromosome, I am getting 2 cells which are containing 23 chromosomes. How many chromosomes are present? 23. So from this one cell, I am getting 2 daughter cells from Meiosis One. Okay, in this Meiosis One, these 2 daughter cells are having half the number of chromosome as that of the parent cell. That's why this Meiosis One is called as reduction division. What do we call them? We call them as reduction division, right? Now, what is the next one? It is Meiosis Two. Now, in Meiosis Two is equivalent to what? Mitosis. There you can find it is an equational division. Like in mitosis we learned from 46 chromosomes 2 cells were formed, right? So here it is 23. So from 23 also how many I am getting? 23 2, and from this also I am getting 23 2. So at last of meiosis you can find how many cells are formed? 1 2 3 4 cells are formed, and all these 4 cells are having haploid number, that is half the number of chromosomes, right? So this is regarding your meiosis. So Meiosis One is a reduction division, Meiosis Two is what? It is an equational division, because the number of chromosome is equivalent to that of the parent. That's why it is called as what? Equational division, which is similar to mitosis. Okay, so this is regarding meiosis. Now we will go in detail regarding the different stages. Listen, so Meiosis One, it is called as what? Reduction division. Meiosis, there are 2 stages, one is Meiosis One and Meiosis Two. Meiosis One is reduction division, 2 daughter cells are formed and the number of chromosome is half the number. From 46 it is reduced to 23. Okay, and Meiosis Two is what? Equational division. Here 4 daughter cells are formed and all are having equal number like 23 as that of the parent cell. Okay, so that are the 2 stages of meiosis. Now first we will be discussing Meiosis One. Listen very carefully, a bit difficult to understand, listen very carefully. Okay, so what is Meiosis One? What is happening in Meiosis One? Meiosis One is divided into sub-stages. What are they? One is prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. Okay, so what are the stages? Prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. Prophase, metaphase, anaphase, telophase we have already covered. Now here some changes. What is that? See, this prophase 1 instead is divided into 5 more stages. What are they? Leptotin, zygotin, pakitin, diplotin, dikinesis. These are the 5 stages of prophase 1. So what is happening in this 5 stages? That is what we are going to discuss. Okay, now listen very carefully what I am going to say. It's very important. Now we are having how many number of chromosome? 46 chromosome, and this 46 chromosomes, when you are seeing, they appear in pair. These pair we call it as homologous chromosome. What do we call them? Homologous chromosome. So this is one chromosome and this is another chromosome. So like this, how many pairs are there? You can find 23 pairs. How many pairs are there? 23 pairs. Okay, so this is one pair, this is second pair, this is third pair, like that. How many pairs are there? 23 pairs. And all these pairs, when you are seeing, one pair, in this one pair, one set I am getting it from my mother and the other set I am getting it from my father. Okay, so I am having 46 chromosome. This 46 chromosome remains in 23 pairs, and all these 23 pairs, when you are seeing, one set of the chromosome I get it from maternal side, that is mother, and another set I get it from the paternal side, that is father. This two combined together, so mother is also giving me a gene which is coding for my height, mother is giving me a gene which is coding for my color, father is also giving me a gene which is coding for my height, which is giving for color. So all these homologous chromosomes, when you are seeing all these homologous chromosomes, these homologous chromosomes are coding for same sequences. So this is one pair of homologous chromosome, means they, what and all genes are present over the same character gene will be present over here also. Now if my father, imagine my father is my mother is my father is tall and my mother is a bit short. Okay, in that case, the gene for tall will also be present, the gene for short will also be present. Now I will be expressing either of them, or sometimes a mediocre, means I may not be that tall and that short. Okay, which one I will be expressing? Most probably I will be expressing the dominant character. What do we call them? The dominant. You have learned monohybrid dihybrid cross, isn't it? So out of the two genes of alleles of a gene, which one is coding for which gene I will be expressing? The gene which is dominant. So like this, how many pairs I am having? I am having 23 pairs. All the genes are present. I am getting the genes from my father as well as from my mother. Some of the genes of my mother is getting expressed because it is dominant. Some of the genes of my father is getting expressed because it is dominant. Okay, sometimes I am different from them. Why, we will be discussing now. Okay, I am not having some characters equivalent to them. Why? That is what we are going to see. Now, is that clear? So always the chromosome remain in pair. One set is from the maternal side and another set is from the paternal side. Okay, so how many pairs will be there? 23 pairs. Is that clear? Right. Now listen, I told you this meiosis, when does it happen? After the whole process is over, like what and all, G1 phase, the chromosome, the protein content, the cell organelles has all increased because all the cells need it, isn't it? G1 is over, so interphase G1 is over, S phase is there. S phase we learned, isn't it? DNA replication is taking place. So homologous chromosome is present. This is also producing its duplicate copy, and this one will also produce its duplicate copy, isn't it? So how many chromosomes, when you are seeing all this 46 chromosome with its duplicate will be present, right? So this is homologous chromosome, one from father, one from mother. During the S phase, this chromosome is also synthesized and this is also synthesized. What do we call them? We call them as chromatid. Now one we call it as what? Chromatid. Okay, now listen very carefully. Similar, mother's copy, isn't it? So this is maternal, mother same copy. These two chromatid we call it as sister chromatid. What do we call them? Sisters. They are similar. That's why we call it as what? Sister chromatid. Now this chromatid and this chromatid. So these are sister. These two are sisters. Why? Because father's same. So it is sister chromatid. Okay. But when you are seeing this one, that is your, one is father's and this one, it is your mother's. So these two chromatids, when you are seeing, we call it as non-sister chromatid. What do we call them? Non-sister chromatid. Right? Clear? I think it is more clear now. Again, once again I will say because a bit difficult to understand, I can understand. So all the chromosomes are lying in pair, one from maternal side and another is from the paternal side. This chromosome we call it as what? Homologous chromosome. Now during S phase, what is happening? They are synthesizing, DNA replication is taking place. Each and every chromosome is producing its duplicate copy. So this one will also produce its duplicate copy, for example. Okay, so this one further I am coloring it out so that you can understand, right? And this is also producing its duplicate copy, right? So this is again I am coloring it out. Okay. Now these homologous chromosomes, when you are seeing this is similar, that's why this is called as sister chromatid. After duplication, what do we call? We call them as sister chromatid. We don't call them as chromosomes, isn't it? And this we call it as what? Sister chromatids. But this one and this one we call it as what? Non-sister chromatids, because this is different from this, right? So this is the thing which you have to note. With this we are moving on to meiosis, the leptotin. Listen very carefully. Prophase 1. It is divided into 5 stages: leptotin, zygotin, packaging, diplotin, diakinesis. I am taking only one homologous chromosome. So this is what I have taken in this diagram. Okay, one pair of homologous chromosome. One is maternal and another one is paternal. Okay, so each with its duplicate it is present. So in the leptotin, actually what is taking place? The chromosomes are getting condensed. In the beginning, how the chromosomes were in the synthesis phase, before in the interphase we have learned, isn't it? All the chromosomes which are present, they will be in the form of a net-like structure. The net-like structures are condensed, and you can find this homologous chromosome with its duplicate copy forming rod-like condensed form and are lying side by side. They are just somewhat near to one another in the leptotin. So like this, how many pairs will be there? This is one pair, 23 pairs. How many will be there? 23. So this is one pair, like this 23 pairs. 46 chromosomes will be there, each with its duplicate. Clear? Now what is happening in the next stage, zygotin? These two pairs of homologous chromosome, I told you, one is what? One is the sister chromatids, isn't it? So this one is the sister chromatids, and this one is what? The non-sister chromatids. This one is non-sister chromatid, this and this. These two chromosomes are coming and lying side by side. They come very close to one another. They lie side by side, and this is called as synapses. What do we call them? Synapses. Pairing is taking place. They are coming and lying side by side. These two non-sister chromatids are just coming and they are lying very much close to one another. This one we call it as what? We call them as synapses. This process is called as synapses. The paired chromosomes, when you are seeing, we call it as bivalent 2. That's why it is called as bivalent. And when they condense much, they will become visible as 4 chromosomes. That's why the 4 chromatids are nicely visible, getting condensed. We call it as tetrad. What do we call them? We call them as tetrad. Okay. So this is happening when in the zygotin stage, pairing is taking place. What is taking place? Pairing, synapses. Okay. And the synapsed chromosome is called as what? It's looking like 2 sets, bivalent. Each, when they are condensing much, it becomes very clear, like 4 chromatids present. Okay, so that one we call it as tetrad. Clear? So this is taking place where? In the zygotin stage. Hope it's clear, dear. So in the beginning, what happened? The chromosome, homologous chromosome, they came condensed and were present. These homologous chromosomes are coming and just touching, so somewhat like very close to one another. That one we call it as zygotin. Now the paired chromosomes are called as what? The bivalent, and they become much visible like tetrad 4. Okay. Now in the packaging, very important process is going to take place. What is that? That is crossing over. What is going to take place? Crossing over. Okay. So what is crossing over? Listen carefully. In the packaging stage, actually what is going to happen is that this chromosome is one set, isn't it? So they are, just to differentiate between these two, like this it was present. Now they will touch one another, synaptomal complex. So synaptomal complex means where pairing has taken place, the region where they are going to touch one another, this region we call it as chiasmata. What do we call them? Chiasmata. So this means the structure somewhat becomes like this. How the structure becomes like X shaped, you can find. So structure will be becoming somewhat like this. Okay, so like this it will be becoming, right? So this is one set and this one is another set of homologous chromosome. Now this, in this region which touching place we call it as chiasmata. In that chiasmata actually what is going to take place is that crossing over. What is going to take place? Crossing over. A segment of chromosome from this is transferred to this, and a segment of chromosome from this is transferred to this. This process we call it as what? Crossing over. One chromosome, non-sister chromatids are crossing over one another. One set is moving this side, one set is moving. Wherever the crossing is taking place. So in 23 pairs are there. In all the 23 pairs, what will be taking place? Crossing over will be taking place. So now when crossing over is taking place, the genes are getting mixed up, isn't it? The DNAs are getting mixed up there. So this is the reason why we are different from our parents. If crossing over has not taken place, either I will be duplicate of my mother or duplicate of my father. But here what is happening? Here crossing means I will be having all the characteristics either of mother or of the father. But here I am having some different characteristics. Every individual is unique. Why? Because of this crossing over. Why? Some of the segments have get crossed over. So the DNA composition has changed, because of which I am representing a different character from that of my parents. So crossing over is taking place where? In the Paquetin stage only you can find what? The crossing over is taking place, right? So the crossing over will be taking place at the junction where the synaptomal complex was present. So there what is taking place? Crossing over is taking place. Now what will happen is that after the crossing over is over, in the diplotin stage, the non-sister chromatids will try to separate from the chiasmata. Okay, they become separate. When they are separating from themselves, you can find only at the region where the synaptomal complex chiasmata they will be attached. The rest of the part will be separated out and they will be moving out. You can find they are trying to move out. Okay. Now, next is what? Diakinesis. In the diakinesis, what is happening in the chiasmata region, the touching region, there also what has taken place? Separation has taken place. One set is moving towards, one set of chromosome is moving this side and another set is moving this side. Like this, what is happening? Diakinesis is over. Clear? I will repeat once more this one so that it will become much clear. Listen very carefully. Prophase 1. So in prophase 1 it is divided into leptotin, zygotin, paquetin, diplotin and diakinesis. One set is from the mother and another set is from the father. In the S phase, duplicate copies are formed. Like this, how many pairs are there? 23 pairs are there, isn't it? 23 pairs. So this is one pair, like this 23 pairs are there. So in the leptotin, condensation of the chromosome from the threads, it is condensed in the form of a chromosome and they are lying coming closer to one another. Zygotin, they will remain in close association with one another. Paquetin, in the region they will touch, that touching region where the two non-sister chromatids are touching each other, that we call it as what? We call it as chiasmata. In that chiasmata, the wall, the chromosome membrane will dissolve and some segments from here will be moving into that side and some segments will be moving into this of the chromatids. So crossing over will be taking place between the non-sister chromatids, and this crossover is responsible for a variation in an individual recombinant. The help of a recombinase enzyme actually it is taking place. Thereafter, in the region of chiasmata, only they will be touching and the chromatids, non-sister chromatids will try to separate out from one another. Like that, they will be visible like an X-shaped structure. Here you can find they are in the form of an X-shaped structure. Thereafter, in diakinesis, the chiasmata also there, from there they will detach and two will be formed. So these diakinesis you can find how many? One pair is present, right? So this is having original copy with the recombinant. This is also having original with the recombinant. Like that, you are forming what? This chromosomes, right? So this is happening where? In the prophase. So in the prophase, the nuclear membrane started disappearing, all the organelles started disappearing, all these changes has taken place in the chromosome, in the 23 pairs of chromosome. Now in the metaphase 1, what is going to happen? Metaphase, I told you, all the chromosome will come and align themselves in the equatorial plate. So like this, how many pairs? This is one pair, isn't it? So this is one pair, so this pair is here. So like this, how many pairs will be there? 23 pairs will be there. Okay, 23 pairs are coming and lining themselves in the equatorial region. Out of this 23 pairs, you can find that spindle fibers will start to form, isn't it? In the prophase itself, spindle fibers will start to form. From the centrioles, the spindle fibers will get attached to the centromere, and in the metaphase, all are aligned in the equatorial plane. In the anaphase, what will happen? The spindle fiber will contract, isn't it? Listen very carefully. Here, when the spindle fiber is contracting, the homologous chromosomes, when you are seeing one set of the chromosome, this set, okay, so only one set of the chromosome, that is very important. This set as a whole is moving towards this side, this set is coming down, okay. So one set is moving up and another set is coming down, one set is moving up and another set is coming down. So how many pairs were there? 23 pairs, isn't it? So how many pairs will be moving up? 23. This is one pair, right? So one pair is moving up, means one pair will come down. Like that, how many pairs are there? 23 pairs. So 23 pairs will move up and 23 pairs will come down during anaphase. Here only one pair, among the one pair, one set has moved up, one set has come down. Like this, how many are moving up? 23 are moving up and 23 is coming down. Clear? So that is happening during anaphase. So spindle fibers are contracting, they are taking one set of chromosome, the whole set, to one side and another one set to the other side. So like that, towards the poles, 23 is moving up and 23 is coming down. Like this, you can find anaphase, 23 chromosome has moved up and 23 chromosome has moved down. So in the telophase, what is happening? Opposite of prophase we have learned, isn't it? Nuclear membrane will reappear. What is going to happen? The nuclear membranes are reappearing, and you can find these chromosomes are again converted into what? Thread-like structures, and nucleolus is formed, everything is done. What is the next step? The next step is cytokinesis. So in the cytokinesis, the cytoplasm will start to constrict. If it is a plant cell, what will happen? Cell plate will form, isn't it? So like this, you can find in the cytokinesis, division is taking place, and the cytoplasm from one chromosome, from one cell, how many cells are produced? 2 cells are produced. In the beginning of prophase 1, how many chromosomes were there? 46. But now in the daughter cells, how many are there? 23. How I told you? Because in metaphase 1, from there, when it is moving in anaphase, one of the pair will be moving up as a whole and another will come down as a whole. That's why 23 will be present in one side and 23 will be present in the other, right? So like this, you can find in meiosis 1, reduction is taking place. What do you mean by reduction division? From 46 chromosome, I am getting 2 cells which are having 23 number of chromosomes. Is that clear? Now what is going to happen? From this 23, again 23 should be produced. From this 23 also 23 should be produced, isn't it? So this step we call it as meiosis 2. What do we call that? Meiosis 2. Hope meiosis 1 is clear, ma. You have to be very careful. You have to learn what is synapsis, what is bivalent, what is tetrad, what is chiasmata, what is synaptomal complex, what is crossing over, what is the dikinesis significance, okay, what is taking place in the metaphase, anaphase, telophase. All these stages are very important for class 12 also. So try to understand the concept of meiosis clearly. Hope it's clear. Now moving on to meiosis 2. Meiosis 2 is again divided into how many? 4 stages. What are they? Prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. Okay. So 4 stages. What are prophase 2? Listen very carefully. So in the beginning, we learned what happened in the meiosis 1. I am getting 2 chromosomes, means 2 cells which are having 23 number of chromosomes, right? So that is only present over here. one set is present over here like this. How many are there in this? How many chromosomes are there? 23. This side also how many are there? 23. From there only I got, from here only this one only, okay, this is only coming. So sorry, from here this one only, okay, so this and this one, right? So this and this one. So 23 is present here and 23 is present in this cell, two sets, right? Now listen, so in the prophase 2 what and all will happen? I told you prophase nuclear membrane will disappear, nucleolus will disappear, spindle fibers, centrioles will divide and they move towards opposite pole, spindle fibers will start forming, right? All these changes will be happening where? In the prophase, right? Again the chromosome which was converted into threads are now converted into what? Rod like chromosome like structures, right? Now in the metaphase what is happening? So in this metaphase you can find the cells like this, only one I have drawn, one set, isn't it? So like this how many sets are there? 23, right? 23 sets are there. So all the 23 chromosome set will be aligning themselves in the equatorial plane. In this cell also 23 will be present, only one I have drawn, clear? Now in the equatorial they will reach and the spindle fibers will come and attach to the centromere. Now what will happen? Contraction, next stage, contraction of the spindle fibers will take place and the spindle fiber centromere will divide. One set you can find here how is dividing. In the beginning, in the Meiosis 1 the whole set was moving, there was like this, they didn't divide like this, isn't it? One set was moving up, one set was moving down, no, not like that. So the whole set, like in the meiosis when we were speaking about in the first case Meiosis 1, the whole set was going up, whole set was coming down, isn't it? But here this, what is happening? This is dividing, one set is moving up and one set is coming down, right? So here one set you can find is moving up. So this is the set which has gone up from this and this is the set which has come down. Here also you can find one set has gone up with the recombinant, the original one has come down. So like this you can find in meiosis. How many are moving up? 23 is moving up, 23 is coming down, 23 is moving up, 23 is coming down. So I am getting how many? It's 23, 23, 23, 23. Sets has moved up and moved down. In the telophase 2 what will happen? The same. Nuclear membrane will reappear, nucleolus will reappear, isn't it? And this chromosome will get converted into chromatin network like structures, right? Spindle fibers will disappear, right? So all these will be happening in this telophase, followed by what? Cytokinesis. The cytoplasm will start to divide and this will, one cell, from one cell I will be getting two cells. From here this cell also I will be getting two cells. So at last how many cells are produced? 4 cells are produced. And all these 4 cells are having how many number of chromosomes? 23 number of chromosomes, isn't it? So why? Because here again reduction has not taken place, equational. From 23 again I am getting 23 number of chromosomes. So in meiosis I am getting how many chromosomes? 23 number of chromosomes. How many cells? 4 cells. 4 daughter cells are produced. Okay. So this is regarding your meiosis, right? So meiosis, hope so it's clear. Reduction division, so it is divided into meiosis 1 and meiosis 2. Reduction division, so from 46 23 were formed. So for forming this 23 prophase 1 divided into many stages. At last 23 chromosomes were formed, and in meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, from this 2 cells again 2 cells were formed. So altogether 4 cells were formed, isn't it? Now we are going to discuss about the significance of meiosis. What is the next point which we are going to discuss? We are going to discuss the significance of meiosis. So what is the significance of meiosis? Meiosis is very responsible for maintenance of chromosome number. Maintenance of chromosome number. Imagine if this reduction division, the sperms or the egg is produced, gametes, for that only meiosis is taking place, right? So this egg cell or the sperm cell, if meiosis is not taking place, then what will be the number? 46 number of chromosome. This 46 with sperm is going to fuse with the 46 with the egg. Then when combination is taking place, it will result in the formation of a new species which is having 92 chromosomes. That should not happen, right? So in order to maintain the chromosome number in the offspring, okay, offspring means what? In the baby, in the daughter or the son, the chromosome number has to be maintained. For that this meiosis is needed, right? So if this meiosis from this 46 chromosome, since it is resulting in the formation of 23 chromosome, that is responsible for maintaining the chromosome number. Zygote which is formed, it will be having 46, right? So if this reduction division is not taking place, meiosis is not taking place, chromosome number cannot be maintained in the offspring. So each and every organism is having a particular number of chromosome that is maintained by this meiosis division, right? Another one significance of meiosis is it is leading to variation in an individual. Variation, I told you, actually what is happening, these homologous chromosome within them, within the non-sister chromatids, what is taking place? Crossing over, isn't it? Imagine if this crossing over is not taking place, whether everyone will be the different from one another. No, all will be copies of their parents, having all the characters of a father or mother. They will not be having some different characters. So variation is possible only because of what? Genetic variability is possible only because of this meiosis. Okay, that is the second advantage. As well as for sexually reproducing organism, it is playing a major role, helping in what? Reproduction. Okay. With this, the meiosis is over and the chapter is also getting over. Hope you have understood the chapter. In case if you have any doubts, feel free to contact me. Okay. So let's wind up for today. Thank you. Let's meet in the next class with a new topic, with a new chapter, biomolecules. Take care. Bye.